Okay, let's make a start. Welcome everybody. Today we have two interesting talks from Maxine Sherman from the Sackler and Nick Dowell. Nick hasn't arrived yet, but he told me in advance that he's going to be a bit late. He's coming from teaching. So yes, if you have any questions, by all means write them in the chat, and there should be time for questions after in between the two talks. And without further ado, over to you, Maxine.
Thank you. Can you hear me okay? Yeah?
Yes.
Okay, brilliant. So today I will be talking about a project we ran in the lab on time perception. It's a model-based fMRI. And I'll only talk about that for about half of the slot. The second half will be about pre-registration of fMRI data and what we learned from this project. So the basic premise of the project was this. So most models of time perception, their primary goal is to track clock time, so veridical time. And they tend to do this via specialist time mechanisms, such as internal clock models would use pacemakers, so mechanisms that literally count ticks. But the problem here is that they don't intrinsically account for context-based distortions of time. So for example, if we're really bored, time goes slowly. If we're having a good time, it tends to go fast. And the purpose here was to test a model led by work for how these kind of distortions can be intrinsically accounted for in a model of time perception. So the idea is that time is estimated from the outputs of perceptual processing. So we do perceptual processing anyway. How that's achieved is a problem for perception people. And the idea is that the outputs that you get during perceptual processing, these are units of time that we can count. So you don't need a pacemaker, you just need perception. And just to try and illustrate this point a bit better, so imagine you've got some epoch here, and these colored squares are salient events, changes, things that stick out in the epoch, if you like. So for example, if you're on holiday, this might be when you had a really good time on the beach, or when you saw a show or something. And you've got lots of salient events, and these events are units that you can accumulate. And lots of salient events means lots of time. So you would rate this not necessarily as having gone faster or slower, but as being a long epoch. And then by contrast, if you've got this epoch here, the same amount of physical time, clock time, you've got not very much stuff going on. Maybe you're on holiday and you were ill. So here is when you ordered a really nice room service, and here is when you watched a movie, but otherwise there's nothing. You would rate this as not being a very long epoch. So this is the proposal, this is the model. And we wanted to test whether this is the case by using functional neuroimaging. So we've got 40 people, and they watched videos, naturalistic videos, either of an office or a city. So the office videos, I didn't put them up here, but the camera is just sitting on a desk and people are working. So extremely boring, almost nothing going on. And then the city videos, an RA was walking around Brighton town centre. So you're seeing changes in the scenery, the new buildings, people walking back and forth, much more interesting. And they varied in length. And this was pre-registered. I'll mention the bits that are exploratory. So we've got about 60 trials per person on average. And so the crucial DV here is people's bias in subjective time. So we weren't interested in the extent to which people could predict time, i.e. Show responses that correlated with clock time. We were interested in the extent to which people were under or over reporting duration. Did it feel long or did it feel short? So then for each person and for each duration category, of which there were 5, we just took people's report on one trial, so for example, 30 seconds, and then subtracted it, it's just mean-centered, subtracted it from their average report in that condition, standardize it. And then we have relative to people's overall bias. Was each video over or underreported? Did it feel long or did it feel short? So when I talk about bias, this is what I'm talking about. Okay, so for the behavior, everyone could do the task. So here we've got the length of the video, and here we've got how long people rated it as. Each colored line is a different person. Everyone's got a monotonic relationship with time. So they are doing the task properly. And if you calculate this bias measure, what it shows is that the city scenes with lots of stuff going on, these were overestimated relative to the boring office scenes. So behaviorally, the more salient events there were, the more stuff that was going on in the video, the longer it felt. So that's good. And then, so the first step was to say, okay, let's see if we can actually generate this effect from the video stimuli, from salient events in the video stimuli. So we've got this convolutional neural network called AlexNet. It's a perceptual classification network, pre-trained, we didn't do anything to it. We just gave it the videos that people watched. And while it's classifying features in these frames, you get node activation at each layer. So these are the layers, these are the dots of the nodes. And you can just calculate frame by frame how much each node activation has changed. And then so for each of these layers here, you can see these gray dots. These are hypothetical changes in node activation over frames, where the x-axis is frames. And so these are kind of what we call events, but the question is which ones are salient. So what we did was used a criterion, and I can go into more detail about this later, but it doesn't really matter how you do it. Use this criterion to categorize events as either salient or not salient. Count them up, and now we have a total number of salient events for each layer, for each video. But this is in some weird arbitrary unit. So we want it to be in units of seconds. So we used support vector regression to just map them onto seconds by mapping them onto the veridical video durations. So we didn't map them onto the reports people actually gave. That would be trivial. We just wanted them in seconds. So that gave for each video a model predicted video duration estimate. From those, we can calculate bias. And again, the classifier did the same thing as people. So salient events and the dynamics of the classifier generate these estimates of duration that are higher for videos with more events in. So this is, so far, was replicating work by Warwick and others before. But our question was, given that we're not this convolutional neural network, can you do the same thing with human brain activity? So now the videos are being watched by people in the MRI. And instead of using node activation in AlexNet, we're using voxel activation in visual cortex. So we've got this hierarchy, just like the convolutional neural net, starting at V1 and ending in higher visual regions. And we can just extract voxel by voxel, TR by TR changes in bold to each voxel. Just add them over the whole voxel, over the whole layer. So we've got for each layer and each TR, the amount of voxel activity that has changed. And again, these need to be categorized as salient or not and regressed onto time in seconds. So it's exactly the same process as with the convolutional neural network. We're just using voxel bold. So quick caveat here. When I said change over TRs, what does it mean? So the pre-registered analysis was the absolute difference between bold at one TR and bold at the previous TR. So the assumption here is that bold represents some raw signal stimulus-driven signal that we compute change on top of. But then we also ran, and this one was pre-registered, we also ran an exploratory one where we don't take the absolute difference. And the idea here was that there's loads of fMRI work suggesting kind of prediction error-like responses in visual cortex. So if bold represents some sort of prediction error, then we can just take it relative to the previous time point. We don't need to do this absolute value. So afterwards, we just refer to these two measures as Euclidean distance, because this is Euclidean distance, and sign difference, so without the absolute value. Okay, so looking at the visual cortex results. So these images are representing data from our exploratory measure. And what you have is, just like every slide before, duration estimates constructed just from human bold in visual cortex. It generates estimates that are longer for city scenes than office scenes. And also, if you just correlate trial by trial, pull everyone together, do trial by trial, you see a correlation between participants' reports and the reports of the model, so the visual cortex time judgments. The extent to which they are overestimating time for a video is correlated. In the pre-registered analysis, the correlation came out, not the bar plot. If anyone wants to know why, I can talk about it later. So the correlation came out on both analyses. This t-test came out only pre-registered. So we also had some control models. This could happen not because of perceptual processing, but I don't know, because it's a general pattern that you see in the brain or something. So we also built an auditory cortex model, but we found nothing. So you could not generate human-like subjective time judgments from auditory cortex. They didn't discriminate between office and city scenes, and they didn't correlate with human reports. And this was in both cases, both pre-registered and exploratory. Same for somatosensory cortex, couldn't do it. There was no effective office versus city, no correlation with human reports. So this is specific to visual cortex. I forgot to mention these were silent videos. So the only modality relevant for processing these videos is vision. And the idea is that that's why we only see it in visual cortex. If they were auditory stimuli, we would expect to only see it in auditory cortex. So the conclusions of the study are that we could predict people's biases towards over or underestimating time from bold activity that's associated with perceptual processing, so only from visual cortex. And the idea here is that it supports a model that intrinsically incorporates the content of what you are judging time for. So you don't need internal clocks, you don't need pacemakers, you don't need any fancy stuff for time. Time perception can just piggyback on perception. Does anyone have any questions about this before I go into the pre-registration section? Maybe it makes more sense to do it this way. Or Chris, would you prefer me to just carry on and have all of the questions at the end?
It's entirely up to you, whatever you think is best, but I could ask a question and there's a few other people. So my question would be, you've got these video types, mm-hmm. city and office based. Yeah. Did you do anything to kind of like equate those in terms of, just the amount of activity that's going on in the videos? So for example, if there's just lots more, contrast changes because there's lots more going on in the city scenes mm-hmm. versus the office scenes, you can imagine that would just drive the visual cortex more, and whatever form of classifier you use would pick up on that.
Absolutely. So we did not, because that was That's the whole idea. So the whole idea is that these changes at, say, low-level contrast changes, because there's more going on, because you're moving, etc. These are exactly the kinds of drivers of overestimating time. So say like the differences that you get in V1 bold that are captured by this algorithm, say perhaps they are tracking changes in contrast and there are more of them in the city scenes, then you would overestimate time.
I see.
Yeah, that's exactly what it's trying to capitalize
on. Let's see what other questions people have. Sam's got a question. Can you expand on why the difference in bias did not come out when using L2?
L2 Euclidean distance, sorry.
Yeah, I've got it here. So basically, what we had pre-registered was, because we only had 60 trials per person, we basically created a super subject. So we standardized everyone's bold, we standardized everyone's reports, we Z-scored them, and then combined them into one big fat subject. Problem was, when we did this, the office city difference behaviorally didn't come out. I think I've got these labels the wrong way around. Yeah, these labels are the wrong way around, sorry. So there wasn't even a behavioral effect to find in the bold data. So because of all of this bizarre standardizing that we did.
I see. Makes sense. Yeah. Cool. Thank you.
Chris, did you have a question?
Really quick, hopefully, clarification question that for the figure where you were showing the bold red blobs sort of signifying, yeah, if you could just go back.
I'm not sure where I'm going. Sorry, I couldn't hear you. I think it cut out.
So a bit, so no. So when you were showing the method for the visual cortex, and so you've got one of your figures that shows the different levels. Yeah, that's right. So in number 3, accumulation of salient normalized events, you've got some events kind of schematically marked in red. So how do you? I couldn't understand why they were in red, particularly like in the middle bar.
So over the x-axis, it's time, so it would be the TRs within one video. And the red line here is the criterion. So it's basically got exponential decay. So what it does is it starts at some starting point and decays until it hits something. And then it would be classified as salient and restart. So because I drew the dots quite big, sometimes it doesn't quite work. So if we take here in layer 2, so it would start here and go down, it hasn't found anything, and eventually it hits almost its minimum. And so it says, okay, this one will be salient. Then it starts again at its starting point, decays, hits something, says it's salient, starts again. It's got a little bit of noise as well. And it just is searching, basically, for something to call salient, reducing the threshold until it finds something. You can do this, I think it was in the 2019 works paper. I think you can do this anyway with a static criterion. It's just, this kind of makes more sense if you're getting bored.
Yeah.
You will start to find less interesting things more interesting.
Yeah, okay.
So I think we should just have one more question because time is running short. So maybe Sophie, if you have a question.
I was just going to say, do you think this can explain the disruptions to time estimation in ADHD? Because if you would process perception information less when you're mind wandering, and people with ADHD mind wander more, but like not all the time. So maybe sometimes they would perceive all the events and sometimes they wouldn't, you know?
So I actually know neither about time judgment changes in ADHD, not much about ADHD, so I'm not 100%. But I would imagine that, yeah, if you've got perceptual disruptions, then you would get disruptions in, or changes rather, in your estimates of time, subjective estimates of time. And in terms of attention, so it's, I think in this form, the model might not say anything, but Warwick did an extension piece with one of the co-authors, Zaf. And you can generate all sorts of interesting attentional effects, including effects of cognitive load on time judgments, I think it was.
Oh, interesting.
Really nice. It's just kind of, when you extend this and also bring in memory, it all comes out. But here we've got the kind of model that's slightly more in its infancy.
Nice, thank you.
Yeah, I think probably we should stop with the questions, because we've got 8 more minutes.
Okay, well, it's less than 8 minutes anyway.
Okay.
It was just a quick spiel on the pre-registration side, because, yeah, so the whole thing was pre-registered, and then we had some exploratory bits in there as well. And just to say that I have a quick overview of the benefits of pre-registration of fMRI. So I'm pretty sure most people here have done fMRI before, probably everyone. Extremely rich data sets, enormous analytic flexibility, extremely expensive modality as well. And so, the benefits of pre-registering it is that you're really checking that you're doing hypothesis-driven research, if you are, otherwise pre-registration probably isn't relevant to you. It's transparent, so when somebody is reading your paper, they know what you really did say a priori and what was not a priori. So for example, with this, we had to define our layers in visual cortex, we had to define our control models. It was so easy for reviewers to say, you only tested auditory cortex and defined your hierarchy that way because it worked out for you. Why don't you do these 55 other analyses? They can see that we had defined these a priori, so it's not just that we were selecting analyses that were convenient for the narrative. And then obviously replication process, so easy for everybody to fool myself with rich data sets. So it's great to think about how you're going to analyze it in advance. But how we did it, particularly for this project, was we started off kind of specifying exactly the hypotheses that we had. It was made much easier by the fact that a lot of the work had already been done in Warwick's NatureCom paper that came before the computational paper. Collect some pilot data, and then that's where we set the parameters of the model, the criterion parameters, pre-processing parameters, optimized the analysis as best we could with an N of 4, and then wrote the pre-registration. It was like 2,000 words or so, specifying absolutely everything. SPM 12, smoothing kernel of, I can't remember what we used, 6. I can't remember exactly the precise statistical tests that we were going to use, what the regression models would look like, exactly how you do the p-values, exactly. It's quite long. You can then obviously just copy and paste most of it into a method section, as long as it hasn't changed. So it's not wasted time. This is time spent, time you would spend anyway. It's just you do it before you have the data. And then after writing the pre-reg, got feedback from colleagues, particularly fMRI people, just to check if there was anything we'd missed. And an example of what might happen when you pre-register in this way is that, so we had this criterion that we used with the parameters, so how quickly it decays, its starting point, its end point. These were based on pilot data. And then when we wrote the paper, we decided to just have a look at what happened if you changed these parameters. So did we just, by fluke, find the perfect parameters? No, we didn't. So this is where we were. Based on the pilot data, we ended up here. Clearly, the best parameters are actually here. So basically it's starting too high up. This is too extreme of a change. Worked out anyway, but you know, without pre-registration, maybe an author would have ended up looking around and deciding on their parameters to be somewhere here, where red means stronger effect. So you can absolutely still find effects, especially if everything's been planned as best as possible, but you won't necessarily find the absolute largest effect size you might find with huge analytic flexibility. So to come up with suboptimal analyses are completely normal. If your result is robust, it shouldn't matter anyway, whether you're doing this kind of t-test or parametric or non-parametric t-test, etc. It really shouldn't matter if the result is there. Exactly how you calculate your measure of interest, for example, bias, ideally shouldn't matter hugely if the result is robust. But that said, exploratory analyses are fine as well. Maybe you find that something that you've decided isn't ideal. You just tell the reader. You tell the reader, this is what we thought was important at the beginning, but actually now, having thought about it more, we also think X, Y, Z is important. And that way the reader can follow your thought process. Mistakes and silly decisions are also normal. Maybe in 10 years, if people still do pre-reg, then everyone will be a pro, but it is normal to either make typos in your pre-reg or pre-register impossible things, which I've done before. Not on this one, I don't think. So get loads and loads of feedback from as many people as possible, because otherwise everyone will have to be understanding, but you have to write in your paper that you pre-registered something impossible or moronic. Sometimes data sets are much richer. And for example, you've got a couple of questionnaires on anxiety, say like 2 different anxiety questionnaires. One thing which is becoming popular, instead of pre-registering one analysis plan, is pre-registering a multiverse, which I think is a really nice idea. So, maybe both questionnaires are capturing the same thing. Maybe you're only going to find effects with 1 out of 2 of your questionnaires. Maybe it's particular items or subscales that are important. So in multiverse, what you do is you pre-register your multiverse. So what are all of the different possible decisions I could make that are meaningful? So for example, analyzing item by item, analyzing subscale by subscale, analyzing questionnaire A, analyzing questionnaire B, all of the different pre-processing parameters I could choose, and then you do all the analyses. And you make a little grid of results, and you simply show people what happens under all of these different analysis choices. And if there's something robust there, even if it's dependent, for example, only on having questionnaire A, then people will be able to see visually, regardless of the pre-processing choices, for example. As long as I was analyzing item by item, I see a result. But it doesn't happen if I analyze the correlations between subscales and bold, hypothetically. But it's really nice, if you're not sure how to analyze your data, it's a really nice way of presenting results, is to just do everything and see if there are regularities there. And then, so writing the pre-registration here took us, I can't exactly remember, but I think a couple of months. But it saves you time later because, for example, as I said before, you've basically written your method section in advance. You've determined all of your analysis plans, most of them in advance, and you can just copy paste. Ideally, you also have the codes written because you've analyzed your pilot data. And then for experiments like GLM, where you're contrasting 2 conditions, you have one DV and a couple of conditions, it's trivially easy to pre-register. You just say, okay, I'm going to do the default pre-processing parameters. This is my DV, this is my condition. I exclude participants on these bases, and you write your document, it's 500 words max. And then you get open science brand points for something, trivially easy, I would say. Okay, thank you. And Warwick Zaf, good luck for that.
Thank you very much, Maxine. Maybe there's time for one quick question, if there's anything about pre-registration before we move on to Nick. Does anyone have a question? Just lots of very complimentary comments, I think. Yes, everyone's very complimentary and rightly so.
So
Thank you very
shall I
much.
stop sharing, yeah?
Sorry?
Thank you. I just asked whether I should stop sharing, but I did it anyway.
Nick, are you around? Oh, there we
Yes,
are.
I'm here. Can you hear me?
Yes.
Oh, good. It's the first time I've used this microphone and camera setup, so I'm glad
Actually,
you
the microphone is not ideal. It's a bit kind of muffled. Don't know if you
Yeah.
could move it nearer to you, maybe.
I think it's standard BSMS issue, I'm afraid. I can probably just try and speak a bit louder. That might help, but I can't do anything about the muffling, sorry. Okay, so yeah, I'll get straight on with it. I'll share my screen first. Okay, all right, is that coming through okay?
Yes.
Great. Okay, so this is just a short update really on the technique that we've been developing to measure blood-brain barrier permeability here at CISC. So we've had this technique up and running in some form or other for a couple of years now, but there's been a recent iteration which has seen an improvement in the quality of the quantification of permeability in the blood-brain barrier. So it's sort of timely that we've got this opportunity to tell you about that here. So I'd start off by just giving you an overview of why you might want to look at the blood-brain barrier, what it is and what it does. I'll go through some of the modeling options of the signal that we use and describe some of the challenges involved, and then give you an overview of the current acquisition and some examples of some encouraging results. Okay, so the blood-brain barrier is poorly named really. It's more of an interface than a barrier, a semi-permeable interface that separates the circulating blood from the brain tissue. So it's constructed of endothelial cells, astrocytes, parasites, and microglia. And a cartoon picture of it here, if you take a lateral section through it, you can see that what it does is it just protects the brain tissue, this sort of orange bit here, valuable brain tissue, from potentially toxic species. So plasma proteins, pathogens, red blood cells, leukocytes, and any circulating metals that we find in the blood circulation. If they were to get into the extracellular space, then this would lead to neurodegeneration. You get neuronal toxicity and oxidant stress. So the idea of this blood-brain barrier is that it only lets through the good stuff, but keeps out the bad stuff. But in certain types of pathology, this blood-brain barrier becomes compromised. So, for example, in cancer, stroke, MS, small vessel disease, or inflammation, we see that this blood-brain barrier becomes compromised and becomes more permeable and can actually let out some of these neurotoxic species into the brain tissue. Now, this damage, the leakage in the blood-brain barrier can be measured without using MRI, and this is typically done by looking at the CSF, a serum blood albumin in the CSF, where things leak out from the blood into the CSF. But to get to CSF in a non-MRI kind of way, you have to go in quite invasively, perform a lumbar puncture to recover some CSF, and then do your analysis of that, which helps you to quantify how leaky the blood-brain barrier is, perhaps. But what it doesn't tell you is where that leakage is taking place, and that might be crucial for a particular type of disease. And these are things that MRI does have the possibility of delivering. So by using dynamic contrast enhanced MRI, you can actually quantify leakage through the blood-brain barrier, but also localize where this leakage is happening. And the goal is to generate quantitative permeability maps where you can actually look pixel-wise across the brain and identify regions in the brain that are more permeable than others. So what exactly is this dynamic contrast enhanced MRI? Well, in a nutshell, it's just repeated T1 weighted imaging. So a bit like fMRI is repeated T2 weighted or T2 weighted, T2 star weighted imaging. DCE MRI is repeated T1 weighted imaging. And you collect lots of T1 weighted images during and after the delivery of a contrast agent. So when you inject someone with contrast, you inject them into their bloodstream, and so gadolinium will pack the bloodstream, as I've got shown here in this vessel. And then some of it will begin to cross the blood-brain barrier, partly because it's semi-permeable anyway, but also it'll start to cross at a greater rate if there's damage, if there's compromise to the blood-brain barrier. And as the gadolinium leaks out, it gets trapped in this extracellular space, and gadolinium causes the surrounding water molecules to appear bright on T1 weighted imaging. So the more gadolinium that leaks through the blood-brain barrier and gets trapped in the brain tissue, the brighter that brain tissue will appear in our T1 weighted imaging. So what we're doing is we're collecting lots of T1 weighted images repeated of the brain and just checking the signal intensity in the brain tissue. If we see it going up, then we know that gadolinium is getting lodged there. And we can track this signal change over time, and the rate at which it increases is an indication of how permeable our blood-brain barrier is in that location. So I hope this comes out on your screen. So this is like an animation of gadolinium entering the brain through the bloodstream. And you can see this bright area is actually the draining vein. This is the central sagittal sinus vein, taking blood from the brain back to the heart. But you saw as it came in, it suddenly flared up as a bolus of gadolinium enters through the vasculature and then starts to drain out. If you were to plot this, but if you put a region of interest over one of the feeding arteries, like the carotid artery, for example, this is what you would see. A lot of stuff goes on in this early period here. But you can see after this early period, things generally calm down and get a smooth curve, but elevated signal intensity throughout. This early stage is composed of a baseline. So typically you start scanning your T1 weighted imaging 20 seconds or so before you inject. So you end up with a baseline of what the signal would have been without the gadolinium. And then it races up very quickly as the bolus of high concentration gadolinium enters. And then it drops as this big bolus of gadolinium flushes through and comes out the other side on its way back to the heart. And then you get a recirculation where the blood pumps it around again and you get this second little peak. But after that, after about a minute or so, things settle down and you get quite a smooth decay of gadolinium as it spreads around the body and starts getting dumped out by the kidneys. So modeling the change in T1 weighted signal as a result of this gadolinium leakage can be done by a range of different models. They can generally be partitioned into 3 different types. 2-compartment exchange, which is the most complete model. Then there's various simplifications. So there's a TOFS model, extended TOFS model, which means you only fit 3 parameters. You have 4 parameters to fit for the 2-compartment exchange. And the most simple model is the PATLAC model, where you just have 2 parameters fit. So if we look at those in more detail, the more complete model, the 2-compartment exchange model, requires us to fit for the plasma flow, this FP, and then the size of the plasma space and the size of the extracellular space, and then the exchange of gadolinium between them. So those are the 4 parameters that you need there. The extended TOFS model just makes the assumption that the tissue is highly perfused, in which case you don't need to worry about the flow rate. It's high enough that it doesn't have an effect on the model. So then you're just left with the concentration in the plasma, concentration in the brain tissue, and this rate of transfer, this permeability surface area that links the two. And here's the equation here that you would then do your fitting on for the data. The simplest approach, though, is the PATLAC model. And this one just looks at the transfer of gadolinium from inside the blood vessel to extracellular space. Whereas the TOFS model, it makes the assumption that some of this gadolinium can get back. You'll build up enough concentration in the brain tissue that some of it actually could start moving back into the blood vessel. The PATLAC model says you're not giving that enough time to happen, or there's not enough gadolinium going through in the first place, not permeable enough to worry about gadolinium coming back in. So this is the simplest approach. And actually this PATLAC model turns out to be the most appropriate for imaging subtle permeability in the blood-brain barrier, because it's so difficult for the gadolinium to escape that you only get tiny concentrations building up in the brain in healthy people, for example, or people with only subtle blood-brain barrier damage. And so you don't really need to worry about it trying to get back into the bloodstream at the end. And the advantage of PATLAC model is it's simpler. You only have two parameters to fit: the proportion of plasma and this rate of gadolinium crossing the blood-brain barrier. So that's the one that we pursued in this study, because we're looking for subtle effects in the brain. So just to push that home, these values here are the transfer rate values. How quickly does gadolinium pass through the blood-brain barrier, K trans? In lesions, this is about 0.2 to 0.3 per minute, is that rate value. But in healthy brain tissue, it's much, much lower. So in the case of lesions, you might need to worry about some of the gadolinium getting back into the bloodstream again. So you probably go for the Tofts model there. But in healthy brain tissue, it's so difficult for the gadolinium to pass the blood-brain barrier. We're using the PATLAC method here. The downside of having this low permeability is that signal changes are very tiny. So if you simulate what kind of signal changes you expect to see on a regular T1 weighted image, you end up with a graph like this that shows a signal enhancement as a result of natural, healthy blood-brain barrier permeability with gadolinium. And you can see the signal intensity after about half an hour, giving it half an hour to permeate across the barrier, you only see a signal enhancement of 1%. So you're trying to identify a tiny 1% difference in your signal intensity. And you can probably keep someone in there for about an hour, in which case you've used up all your scanner time and you've got about a 2% increase in signal intensity that you're trying to get. So it's not ideal. There are some big challenges there. And it's made all the more difficult by the fact that the machine is not particularly reliable in terms of signal intensity. Signal intensities, you would like them to be identical each time if nothing was changing in your participant. But unfortunately, you get something called machine drift, where these signal values, pixel values, start changing in a way that's very difficult to understand. The MRI scanner is a very complex machine, and so you see some very odd signal changes over time, probably due to thermal changes within the different components in the scanner. But given that you get given the scanner in a really different condition each time, someone might have been doing fMRI before you, when you start doing your BDB measurements, in which case you have a different thermal starting point than if you were scanned at the beginning of the day, for example. So trying to estimate machine drift is incredibly difficult. Then you've got the problem with people actually moving in the scanner. You're doing this repeated imaging, and yeah, you can co-register, but you always end up with problems of co-registration artifacts, or because the received sensitivity, the signal sensitivity changes in a spatial, is spatially dependent. If they move their head to a slightly different place, they might get different signal intensities just by the fact that they've moved their head. And given that you're only trying to measure 1 or 2% differences, this can really screw up your quantification. So picking an acquisition scheme that minimizes all these effects is absolutely crucial. And one of the first approaches we took was a very gentle approach, which by gentle I mean we were stressing the machine only slightly by acquiring very slow imaging, a very long TR, trying to make sure we don't warm up the scanner too much, trying to avoid any signal drift. So you can see over time here, we were scanning someone. We didn't give them gadolinium at this point, so we expect to see no signal change at all over time. We were scanning them, and it took about 3 or 4 minutes to collect that data point, and then we waited a bit, collected another one and another one. And you can see we get this massive signal drift of 2% that occurred with someone that didn't have any gadolinium. It should be the same signal throughout the entire scan. But over 40 minutes, we see this bizarre behavior. It goes down a bit, goes up a bit, and then does this sort of sinusoidal down and then upward tilt. Quite what's causing that is anyone's guess. It's a very complicated problem to try and solve, and no one has solved it, and I doubt they will. So that was the original protocol, and that resulted in some very hit and miss measurements of this K-trans permeability value. You could only really do it on a region of interest by taking a collection of pixels in an ROI. And even then there were some failures. So these circled points here are showing no permeability when we're expecting sort of normal healthy permeability in this region of the hippocampus, it was here, to be about 0.5. But it's a bit low and sometimes it completely disappears just because it's getting lost in the noise of all of this machine instability and potentially subject movement. So we introduced a new technique more recently. So this is the original protocol here, just by way of reference. But the new protocol, we transitioned from using a gradient echo, simple gradient echo, to this vibe, which is another gradient echo technique, but it's much quicker. It allows you to collect many more volumes in the same amount of time. So the TI drops from 30 milliseconds to just a couple, which will reduce your signal intensity because you get more T1 weighting with that short repetition time, which is why my voxel sizes here have increased to try and recover some of the signal intensity. But we do have much more rapid imaging, so each volume is about 2.5 seconds compared to about 1.5 minutes with the old technique. And this brings down our total acquisition time. So this then allows us to start using that PATLAC method now, because we're acquiring data in a much shorter period of time, where we don't need to worry about gadolinium making its way back into the bloodstream. So what are the consequences of moving to this new technique? Well, this is your signal to noise ratio comparing the new technique, the red one, vibe, with the old gradient echo. And you can see we really get punished by using that short TR. We get greater T1 weighting, which reduces our signal greatly. Like I said, you can recover some of that by increasing your voxel sizes. So each pixel represents more hydrogen nuclei, so you get greater signal intensity. But signal to noise ratio will always be lower in this more rapid imaging. But the big advantage, especially in the context of trying to measure really subtle changes in T1, is that you're sensitizing your signal to T1 much more. It's got a much greater sensitivity and a much greater contrast to noise ratio. So here is the sensitivity, the signal change as a result of an increase in 1% of T1. So gadolinium leaks out into the brain tissue and causes a T1 change of 1%. It'll cause our vibe signal to go up almost 9-fold. Whereas with the gradient echo, it's like 2 or 3%, right in there with all the machine drift problems. So we've killed a lot of our signal, but assuming you've got enough to be able to detect changes, you're much more sensitive to smaller T1 changes. So what does this look like when you actually come to collect the data? Well, this is my protocol here in cartoon form. So prior to the vibe acquisition of the BBB, you have to get a good measure of T1, because that's in part of the model. You need to know what the T1 is before you start injecting anyone with gadolinium. So you need to do good T1 mapping, and also B1 mapping is advisable as well to correct for the corrupted B1 effects. So you do a bit of pre-scanning that's required to correct for some of the problems later on. Then this is followed by, in this case, I did 432 brain volumes with a time resolution of 2 or 3 seconds, and injection of gadolinium 20 seconds into the acquisition. So 20 seconds worked out about 10 baseline images, and the gadolinium goes in, and then you observe what happens to the signal intensity after that. And then you fit the data using the PATLAC model, which I've incorporated into a sort of friendly GUI in MATLAB, which allows you to do this. You select your region of interest and then measure the signal intensity change over time. And then this reformats it into what's called a PATLAC plot, which you then read off the slope to get things like the K-trans, this permeability value. And we really think that this approach is much better than the old approach. So I've done a comparison of the two, looking at signal variability. So there's many fewer data points here with the slow technique, because that was 90 seconds per data point. So if you look at just the standard deviations alone, it looks like the gradient echo is a bit better. Here it's a bit noisier because you've got your lower signal to noise ratio. But when it comes to fitting for the blood-brain barrier, you use all of the points. And what you're really interested in there is the standard deviation of the mean, the standard error. And it's much lower for the Vibe technique because you've got many more points. So when you fit your data across all of these points, you end up with a better, more reliable value for the signal change. So these two sets of data you just saw there were acquired without any gadolinium. So in an ideal world, this would just be a straight line on 0, showing no deviation at all. But you can see that there's drift, and there's drift in both cases. So we get drift with our slow gradient echo, where we're trying to be really kind to the scanner with its really long TR values. And drift, is 3% or so. And we see exactly the same drift for the Vibe over a much shorter period of time. Notice after about 15 minutes or so, we end up with that 3% drift. It took like 30 odd minutes to get that same drift with the slower gradient echo. So the drift rate is lower here, but the overall drift across the whole data set is about the same. So you don't really get penalized by pushing the scanner really hard here, because you're acquiring for a shorter period of time anyway. And there are ways that you can account for drift. You can, for example, here, this is someone's head here, the side of someone's head. And just on the outer side of the coil, we've sellotaped a test tube full of a gel with a known T1 that we can measure. So we can measure this signal intensity. It shouldn't be linked to physiology or gadolinium being pumped into this person down here. So it should remain stable throughout the entire scan. And then we can use any drift that we see in this signal intensity to try and characterize and correct for any machine drift. But this does have some problems associated with it, because the intensity of this phantom here, this test tube of gel, will be dependent on its temperature. So if there's a fluctuation of temperature in the room, this participant warms up the coil and warms up this phantom, then we'll see a drift due to temperature. So it also opens up a potential avenue for more variability that's corruptive to our data set. Like I said, the receive sensitivity of our head coil is quite appreciable. So this is just an image of someone's brain here, no gadolinium or anything like that, just a regular image, but I've changed the contrast to show you that the bits of the brain that are near the receiver coil, so the front of the head here and the back, appear much brighter than those buried deep within the brain. And this is because it's closer to the receive elements and therefore appears brighter. And this is really problematic for blood-brain barrier imaging, because if someone moves, then the pixels here that appear brightest will change in the brain. If they move their head closer to the coil, it'll get brighter. If they tilt them away, it will change in a difficult to predict way. But what you can do is you can measure what this B1 field is and try to correct for it. As you observe them moving during your co-registration, you can try and correct for any B1 changes in the images. Another problem is image artifacts. So here are two images, pre-gadolinium and post-gadolinium. And you can see post-gadolinium, we have this really bright vein here. And this brings with it lots of image artifacts, which you can't really see here. But if I turn up the brightness like this, you can see it's really bright in the brain now, as you'd expect. But what we're really looking at is the noise. So there's a bit of artifact in the noise. This should be just purely black here, because it's just air. But you can see there's a bit of noise in there from the scalp and the edge of the brain. But once gadolinium goes in, then you have this really bright vein, which causes all of this extra signal intensity, which spreads out into the noise, but of course spreads out into the brain as well. So you've got noise lying across the brain, which have this corruptive effect. You might be able to, and one of the things we're looking at is the possibility of trying to limit this by applying saturation bands that might attenuate some of this signal and reduce some of this effect. But in spite of those challenges, we've seen some really encouraging results. So here's some, like the holy grail really of BBB, permeability imaging, is to try and get a map for permeability across the brain. And this is what we're able to generate using that faster vibe technique here. So this is the K-trans value here, going from red, where it's low permeability, up to high permeability in the whiter areas. And this is a pixel-wise mapping that we're able to achieve that can identify different areas of permeability, which by and large agrees with what we expect from the biology, and agrees with the kind of imaging that we see from the literature that have implemented this technique. Although you will notice there are regions of the brain which do fail. So these areas here in the cortex that aren't colored are because the K-trans mapping was corrupted by all those things that I mentioned, principally because it's at the edge here, edge artifacts from that vein that comes over the top of the brain. And you can see this is a published paper a few years ago now that does the same thing, that tries to do this mapping. And again, you can see that they have problems in the cortex here and really high values at the edge, so edge artifacts occurring. But if you can do pixel-wise mapping, you can also do region of interest mapping, so just looking at individual regions. And you can see when we compare some of our principal regions like the hippocampus and the white matter with those in the literature, they lie well within the range. But you'll notice that this range is massive. And if you look around the literature, you see really different values for permeability K-trans in any particular region you care to look at, which suggests that this technique for working out K-trans is highly dependent on the way that you implement this technique. But what's important really is that you get something that's reproducible at your particular site, as with so many of the things with quantitative imaging. But this technique is still sensitive to artifacts. So that image I showed you before was one of our better participants. If we scan someone else who had a bit more movement and a bit more image artifact, you can see more bits of the brain are not fitted here. And some of those within the brain also are probably not that reliable as a result. So you still have to take care of things like B1 mapping and B1 correction drift. Even though we are more sensitive to the detection of gadolinium leakage here, we still have these problems that need to be addressed. So these images, all the images you've seen, haven't gone through any of these drift correction or B1 correction yet. But that's the focus of the ongoing work. So it's a work in progress. We need to reduce some of the image artifacts. So what we probably end up doing is lengthening our repetition time, because this is a good compromise that will allow us to get the same sort of standard deviations of the means that we're seeing with the vibe technique, but hopefully without the artifact that could cause some of these edge effects. Also looking at how to maybe apply saturation bands to reduce high signal from the scalp and the blood in that vein, and then applying a robust method for B1 correction and drift correction using phantoms will all be advantageous, whilst hopefully trying to avoid the introduction of any other errors. So, I mean, we'd really love to have a reliable BBB measure, and we can do that. And I've shown you can actually, if you have a good subject and everything's in your favor, you can get good K-trans maps in less than 20 minutes at the moment using this technique, using this vibe technique, which has this lower standard error and this improved sensitivity to T1 change. But still, there are further optimizations that are required and possible. I mean, this is slowed a bit by the fact that you need to inject people with gadolinium. So the whole process, from an ethics point of view, means that the development is necessarily slow and non-stop, and is not possible at the moment anyway because of the whole COVID thing. So there's been a bit of hiatus on that. But hopefully, I've convinced you that things are heading in the right direction, and we're getting to the stage where we've almost got this reliable blood-brain barrier permeability mapping in the brain. So none of this stuff would be possible without my co-workers, and especially Jenny Rusted and our PhD student, Neuro, who was involved in collecting a lot of this data, and who helped Jenny helped us secure the funding in order to perform this sort of physics side of this development, which is now being rolled out on people with different ApoE genotype status. So the hope is that in a few months' time, Neuro will be able to show how this technique has been applied to these different genotypes to detect differences between different healthy volunteers with the E3 or E4 variant of ApoE. Thank you very much.
Thank you very much, Nick. We are pretty tight on time, but I guess people can leave if they wish. If there's any questions, does anyone have any questions? Sam had a question saying, does that mean that the Patlack model cannot account for the decay in the signal? I think maybe you might have answered that over the course of the talk. But,
yes, yeah. So it's modelling the increase in gadolinium into the brain tissue. The actual loss of gadolinium for the brain tissue is a much slower process. So it's a low concentration anyway, and the chances of a gadolinium finding its way out is very slow, especially over the time period of the scan, 17 minutes or so. So it becomes an issue if you do an hour worth of scanning, like with the old technique, which is why the extended TOFS model would be the way forward if you're scanning for an hour or so, because there is more chance of gadolinium finding its way out. But in general, that's not a problem with the shorter implementation of this. So the Patlack model is the way to go, because you've got one fewer parameter to try and fit, and that can screw up your K-trans by trying to fit that extra parameter, which is very low anyway.
Cool. Thank you.
Is there any more questions?
May I ask a very cheeky one, additional one, if I may? So, do you think that the drift is caused by heating of the scanner itself, or could it be heating of the participant's brain? Which is it more likely to be? Because I see this drift in our fMRI data all the time, and it's annoying.
Yes, and I think that it's confirmation that it comes from the scanner. So with fMRI, typically you're zapping them with single RF pulse. You give them a 90-degree RF pulse. So the SAR, if you look at the SAR value on the scanner, if you do fMRI, it's tiny, a couple of %. So you're not heating your participant at all with fMRI. It might change if you start doing multiband and you start packing in the RF pulses. But if you've got relatively long TRs, then the heating of your participants with EPI is really slow. So it all comes from the scanner really, and it's just really difficult to work out where it's coming from. There's lots of different components, and it's very difficult to pin down. I mean, I had a plot that showed how I scanned overnight once. I set just put a phantom in there and left it overnight, scanning from 6 o'clock in the evening until like 7 o'clock the next morning. And you see this sort of exponential drift occurring in the scanner on that particular occasion. At the beginning of it, it drops by like 1 or 2% in the first hour, and it sort of gradually tails off. But it's you do get appreciable drift throughout the night. And if you wanted to get to that flat bit of the curve, you basically have to be scanning for like 5 hours, and then you can get to a regime where the scanner is sort of not drifting that much. But I mean, that's not practical. I mean, I guess you could make sure you're the first people to scan in the morning and have the scanner just pulsing away, doing this monotonous gradient echo repeatedly on a phantom, then quickly rush someone in there before it changes. That might be a way to do it. Might be worth trying doing that actually. But it's impractical, and but I don't pay the electricity bills at CISC. So.
Thank you.
Thank you very much, Nick, and thank you everybody else for joining us. I think we should probably stop. Next week we have some project proposals. So I look forward to seeing you all then. And thank you again.
All right.